patient Tracy walks into a Texas hospital. This was back in the 1990s, I believe, maybe even late 1980s. And patient Tracy, does anyone know the study or this story? Oh. Patient Tracy had LDL levels in the teens. So, Lillian, you're more familiar perhaps with this data. I mean, this is unachievable, right? This is sort of off the charts. This is a data point maybe we would throw out of the data set because it's one of those outliers, right? And so, is this real? Maybe if, a student or if I, as a trainee, presented my PI with this data and I said, well, there's a 14 for LDL. Do you think that's real? Maybe not. But in this case, it was. And so normal target levels for LDL are more commonly around 100 milliliters per deciliter, a level that many people fail to achieve, 100. So, Tracy is African American. She and a handful of other African Americans share one thing in common, the genetic mutations in the gene PCSK9. Tracy inherited mutations, one from her mother and one from her father, which eliminated a protein in the blood that has a fundamental role for controlling LDL cholesterol. So researchers were fascinated. Why does Tracy have this? And it turns out that African Americans with similar mutations have a 90% reduced risk of heart disease. Can you hear sort of the pharma companies like cashing, thinking about how much they're going to cash in on this finding? So Helen Hobbs, an MD, Jonathan Cohen, a PhD geneticist, and Eric Borwinkle, who is still very active in research, actually was just on a call with him the other day, a genetic epidemiologist. So they all found out about this finding and worked with the clinicians who were seeing patient Tracy. They did a battery of tests on Tracy. No one understood what was going on with Tracy. And Tracy's just healthy, as can be, walking around with an LDL of 14, right? Living life. And so, maybe not so much after she was pricked and prodded. So she should probably have benefited from this actually financially, if you ask me, but who knows. So anyway, identified PCSK9 as a gene of interest and explored associations with CVD outcomes. So they had this Dallas Heart Study, right? They were investigators on this Dallas Heart Study, and they say, okay, we found PCSK9. It's associated with low LDL levels in Tracy, and we are now seeing this in other African Americans. And they had this cohort study of about 3,500 people of African Americans and Caucasians, right? So now we're being case control from the Dallas, Texas metro area. And what they found was that they identified the highest and lowest LDL readings in 4 groups. So they took the extremes. And they looked at the extremes of LDL in black women, black men, white women, and white men. They found 7 African Americans with 1 of 2 distinct nonsense mutations in PCSK9. So these mutations aborted the production of the protein. And when they went back to look in the entire Dallas Heart Study population, they found that 2% of all black people in their study either had both mutations, and each was associated with a 40% reduction in LDL cholesterol in the blood. And so here are their findings. This is actually a combination of findings that was reported for the lipid and metabolic meeting not too long ago. PCSK9 and LDL, the iconic example of translational medicine in the genomics era, because they were able to take from discovery, right, in a patient, a nonsense mutation that caused low LDL levels. They took it to the population-based data, where they were able to interrogate an entire population data set to look for individuals that also carried this mutation. And then PCSK9 inhibitors came on the market. So they target and inactivate a specific protein in the liver, and this knocking out the protein dramatically reduces the amount of harmful LDL cholesterol that is circulating in the bloodstream. Lower LDL translates to healthier arteries, fewer heart attacks, strokes, and other problems that are related to cholesterol. So here's the drug now that, based on this PCSK9 inhibitor data, can help you lower your LDL. So when did I say that patient Tracy walked into the clinic? Early 1990s, maybe late 1980s, right? This paper was published last year on the implementation of a new clinic-based pharmacist-managed PCSK9 inhibitor consultation service. How many years between discovery and implementation of evidence-based findings are there? So this is part of the problem, right? Is that there is a huge lag between discovery and implementation. And I think that there's a paper recently that came out that says that on average it's about 17 years. Somebody argued that's arbitrary, but I actually, I was in a meeting last week at NHLBI and they were arguing that that was arbitrary, so I don't know. But I think on average that makes sense. So 1999, the Dallas cohort study was initiated. This is even after patient Tracy had walked into the clinic. 2002, the genomic assays were performed. 2004, results were published from the study. 2015, FDA-approved PCSK9 inhibitors. And then 2018, you're exploring the implementation of this evidence-based therapy. So where are we with precision public health or precision medicine? I think we're a ways away. All right, so this is where my work comes in. And let me just check time. So we're okay, I think, so far. So this paper was published in 2014. This was right around the time that I was starting my faculty position. And Williams and others who were part of the Sigma Type 2 Diabetes Consortium published this paper on SLC16A11 and type 2 diabetes. So this is a gene, it's a solid carrier gene. It's sort of involved in like the transport of cholesterol in the cell, actually the macrophage specifically. And they said, if you have these 5 variants within this gene, and you're Mexican, because this was done in a population of Mexican individuals from Mexico, you're 30% more likely to have diabetes. I think the exact number is about 26%. And so I thought, gosh, I have access to this data which has these 6 different background groups of Hispanic Latinos, right? The SOUL study that I mentioned that has the 6 different background groups. Why don't we look within SOUL to see if we can replicate these findings in the Mexicans and maybe the other background groups of Hispanic Latinos? And so that's what we did. So the Hispanic Latino Community Health Study, the Hispanic Community Health Study of Latinos, took individuals of Hispanic Latino background, recruited them from Miami, the Bronx, Chicago, and San Diego. This is a description of the different groups that are in the sample, including another category which we did not analyze, and all of the different PCs for heterogeneity. And so you see that there is, in fact, a lot of heterogeneity even within the background group of Hispanic Latinos. And the ancestry comes from all over. So they have some proportions of European, some proportion of ancestry from the Americas, Native American actually, and then some proportion of African ancestry. And I wanted to know, do the associations of the 5 SNPs with type 2 diabetes status replicate in each of the background groups of SOUL? And is there evidence of group-specific effects in each of the groups? And so these are the results from the Williams paper, so the original paper. So these are odds ratios, and anything to the right side of the 1 is what? So there's an association with like maybe increase for disease, right? Or association with type 2 diabetes. Anything to the left is preventive. And you see that for all of the groups. So the initial scan happened in the sigma T2D cohort, which was all Mexicans, and they're all associated with an increased risk for type 2 diabetes. They did a replication in all of these groups, African Americans, Europeans, these were all different cohorts actually. And you saw some associations in Native Hawaiians, East Asians, Singaporeans, Japanese Americans, and of course here are the Mexicans, but some of these cross the 1, right? So those are non-significant. Might have been due to small sample sizes as well. Nevertheless, there was a statistically significant association between these variants and type 2 diabetes in the Mexicans. So we ran this analysis in Seoul, and we found that we were able to replicate the findings in the Mexicans only and not in the other background groups of Hispanic Latinos, which was interesting. We actually have a paper under review, second resubmission with Nature Scientific Reports, where we're unable to convince the reviewer that these results are robust. The reviewer is arguing that we ourselves also have really low, so that we have a small sample size, and perhaps aren't powered to detect these associations. Whether or not that's the case, based on the work that we did, I'm not convinced that it is. But I don't know. So we're in the process of trying to figure that out. That said, I still think it's interesting that we were able to replicate in the Mexicans only and not the other background groups of Hispanic Latinos. So this is just repeating a lot of what I have said already. There is additional work that I'll talk to you about in a second that suggests that, even with the results that we have so far, this does sort of point at the genetic underpinnings of type 2 diabetes in this population and may have implications therapeutically perhaps for all populations, but we do need to study this in other populations to establish that. So Jose Flores and others at either BU or Harvard published this functional paper recently, and it's also on the same variants within this gene, also in the Mexican population, which basically shows that it functions through 2 distinct effects, so decreasing expression in the liver and disrupting a key interaction which reduces cell surface localization. So, in brief, SLC16A11 could be a therapeutic target for type 2 diabetes, and this work is contributing to that. And so we're, I think, on the verge of something kind of exciting, but we need additional work to make it truly exciting. So when I was in this implementation science program, I thought, okay, so Helen Hobbs and others, it took them 17 years. So how long is it going to take me in my work to get to the point where we're at a therapeutic target and at the point of intervention? All right, so what are some of the arguments that I have heard with respect to precision public health or precision medicine? And the thing that I hear more so over and over is this medicalization of race. Right? I actually hear this argument a lot with respect to race and genomics. I think a lot of people think about discrimination. I think they think about in the Hispanic Latinos, and actually this is the case in Alaska Natives as well, and Native American populations, where people don't want to know what their ancestries are, right? Especially if they're different than what they thought they were. Because I think that has important social and cultural implications that those populations might not be ready to deal with, right? And so investigators coming and doing genomic studies, publishing these findings. Actually, there was a huge lawsuit at the University of Arizona. I don't know if you're familiar with this, but also dealing with type 2 diabetes. Investigators went to one of the reservations and convinced Native Americans from one of the reservations, I cannot recall exactly the tribe at the moment, to participate in a type 2 diabetes study and collected blood. The blood was stored at the University of Arizona. The blood was then used for other studies such as psychological studies and Alzheimer's and so forth. And a PhD student, I think it was sitting in a room like this one day listening to a seminar where they were discussing the effects of, whatever, Alzheimer's research or and using samples from this population. And so she went back to the tribe and said, they're using our ancestors' blood for purposes other than what we were told it was going to be used for, and it became a huge bioethics example. Because we're proximal to Tuskegee, I've had the fortune of going and listening to a lot of the examples and people who participated in the case. And so it led to a lot of revisions in how informed consent is conducted and the way that it's written so that people know what they're doing when they're contributing genetic material. So this particular case was a little bit different. So case number 1 on the medicalization of race is an 8-year-old boy who was phenotypically European, presented with acute abdominal pain and anemia. The body temperature was only 37.9 degrees Celsius, but surgery was considered anyway. And the technician found red corpuscles with hemolytic characteristics on a smear, and so then surgery was canceled. That's what it says at the bottom. So it was canceled after they found the results of sickle cell preparation, right? The child was treated for previous undiagnosed sickle cell anemia, a genetically inherited blood disorder. So it turns out that his parents were from Grenada and were of Indian, Northern European, and Mediterranean ancestry. So phenotypically, the child presented one way, right? And biased, if you will, the medical examiner, the physician, whomever it was that saw the child. And so an assessment about treatment was made based on that phenotypic assessment. And so when people think about the medicalization of race, I think they think as well that if this were more widespread or more prevalent, that there might be serious issues with medical treatment, in addition to what I think people already experience. So, barriers. One of my other interest areas is MODY, which is mature onset disease of the young. And this is a type of diabetes that occurs. 80% of MODY cases are misdiagnosed as type 1 or type 2 diabetes. So MODY is neither type 1 diabetes nor type 2 diabetes. Average time to correct diagnosis is 8 years. So from the time that someone comes into the clinic with MODY, they're either diagnosed as a type 1 diabetic or a type 2 diabetic and not accurately diagnosed as a MODY patient. But it's only with genetic testing that individuals are recognized as a MODY case, if you will, right? And there's even different types of MODY. So there's 4 different types. They all require different treatments. So if you're a MODY patient and you're diagnosed as a type 1 diabetic, you might be put on insulin unnecessarily, maybe. Or not given any treatment, which would work only if you're a GCK MODY individual. So genomics may explain some population differences in disease incidence. So sometimes they're aligned with outcome disparities and sometimes they're not. And so if we study these in isolation, they may obscure important social determinants of health that may have larger contributions to disease. And so having genetic information in and of itself isn't helpful. Having genetic information alongside some of these other data, I think, are, which is what I think precision medicine should be. It should not be just genetics. It should not be, just providing your genome and deciding what drug fits best. I think precision medicine a more inclusive view of precision medicine should include an assessment of social determinants of health. It should include an assessment of psychosocial and cultural factors as well, if we were to have all of that data. So the intersection between precision medicine, genomics, and health disparities gives everyone the best chance at good health. Ideally, it would improve outcomes for communities with disparities in health and would help us to understand the complex interplay that creates these health disparities to begin with. So future directions. So this is what I think should be included. And I actually add this other sciences. This came from International Journal of Proteomics, but I really think that it needs to be everyone in this room and other people in other rooms coming together and saying, what are things that are important for establishing health? And sort of harmonizing all of that data to arrive at the best treatment for that individual. Now, it's hard enough to provide treatment as it is in the 5 minutes that we see practitioners, right, with the information that's available now. But I think studies like All of Us, have any of you heard of All of Us? So that's also an NIH initiative, and they're basically taking individuals and collecting biological samples for whole genome sequence and genomic analyses, but also assessing a battery of surveys and tests to collect information also on social cultural data and geographical data and a whole cluster of variables that may contribute to a more comprehensive approach to understanding health outcomes. So one of the ways I do this is in epigenetics, or in genomics, is by studying epigenetics. And epigenetics is the study of changes in organisms. I study